website. Hello, everyone, and welcome to the Graduate School One Health Emerging Infectious Diseases webinar series. I am Paola Gamboa, Education and Digital Manager of the Graduate School. Thank you for joining us for this new session in our series, One Health Research in French Amazonia, led by Professor Loïc Epelboin. We are very happy to have you with us today and glad to know you are joining us from Algeria, Brazil, the Democratic Republic of Congo, Gabon, Hong Kong, the Netherlands, United States of America, French Guiana, and mainland France. Today, we are very happy to welcome Professor Loïc Epelboin. On March 18, we will welcome Professor Jean-Bernard Duchemin. On April 22nd, we will welcome Professor Guillaume Audin. On May 18, Professor Antoine Adeny. On June 24th, Professor Paul Letournier. Remember that all our sessions take place at 12:30 p.m. Paris local time. A few practical notes before we begin. The 40 minutes presentation will be in English, but please feel free to ask your questions in English or in French. Remember, we count on your active participation in the chat anytime during the talk or during the discussion period. The videos of the previous sessions are already available on the Graduate School website in the Media section Webinar Series entry. I will be sharing the link with you later on. And now, getting straight to the point, I am pleased to introduce Professor Loïc Epelboin. He is an expert in infectiology and epidemiology of infectious and tropical diseases. He completed his medical training in Paris at Sorbonne University and Pitié-Salpêtrière Hospital before settling in French Guiana in 2014. He is a full professor and hospital practitioner in the Infectious and Tropical Diseases Unit at Cayenne Hospital, Centre and Rosemont in French Guiana. And today, he will share with us the results of 20 years of One Health research on Coxiella burnetii infection in French Amazonia. French Guiana has one of the highest reported incidences of Q fever worldwide, making local research essential to understanding transmission patterns. Studying the disease helps identify environmental and animal reservoirs specific to the Amazonian ecosystem. This research supports better prevention, diagnosis, and treatment strategies to protect the population. Before further delay, I want you to enjoy the session. Professor Epelboin, I'll hand it over to you.
Thank you so much, Paola. Thank you to all to be there today. And if you have any question in French, don't worry. Paola will translate to me in English because my French is not so easy. I will talk today about a very interesting point of the French Guiana epidemiology, and it's about Q fever. And when Paola was talking, I realized that the first publication about the phenomenon of Coxiella burnetii infection in French Amazonia is not 2006, but 96. So today I will talk about 30 years of One Health research on Coxiella burnetii infection in French Amazonia and not 20. And obviously, there are some studies I participated to, but some of them are from many of them are from other researchers, and I will show you, especially the Marseille with the Reference National Center of Q fever and rickettsy of Marseille. They made many studies for the 30 years in French Guiana. So just a small reminder about French Guiana, because we will talk about French Guiana more and more, and there are some aspects you get to know. You know that it's a French territory in America, and it's many people think that it's an island in the French Caribbean, but not. It's the only territory on the continent, the only overseas territory, French overseas territory on the continent. All the others are islands all over the world. And we are between the huge neighbor Brazil, and the small other countries of the Guyana Shield, which are Suriname, the former Dutch Guyana, former British Guyana. It's important to understand why we are at the top of the research in French Guyana. We've got only 300,000 inhabitants, but as it's a French and a European territory in South America, we've got some European resources to support people in the hospitals and to make some research. That's why there are so many publications and papers from French Guyana. 86,000 square kilometers, half of French territory of continental France, and most of the territory is covered with Amazonian rainforest. The population is mainly on the shore. And important to know, because I'm going to talk about that, there is a very high cultural diversity. I will talk to you about the influence of the ethnicity in the research on Q fever, but this is one of the points for many subjects of research in French Guyana. There is also a high biodiversity, and as we're talking about Q fever, which is a zoonosis, it's important to know that Amazonia and French Guyana are one of the hottest spots of biodiversity in the world, more than other tropical forests, such as forests in Asia or in Africa. And so the contact between humans and high biodiversity is one of the explanations of the zoonosis we've got here. Let's talk about Q fever. Q fever, the history starts in Australia in 1935 with the investigation of some unexplained fevers in a slaughterhouse in the Queensland, in Brisbane, in Australia. And Professor Derrick investigated this small cluster. And as these fevers, these feverish syndromes were quite mysterious, he calls this fever the query fever, the mysterious fever, query fever, which gave after that Q fever, which is still the name is given to the disease today, due to the bacterium Coxiella burnetii, which was then in the 30s and 40s discovered and then characterized by some Australian and US scientists, such as Professor McFarlane Burnett and Professor Cox, who gave their name to the bacterium. The epidemiological cycle is typical of a complex cycle between wildlife, domestic animals, and humans. There is a wildlife cycle with exchanges between many species of mammals and some ticks, also tick species. And the exact role of the ticks in the epidemiological cycle of Coxiella burnetii in the wildlife is not really well known. And you get to know that probably many publications on Coxiella burnetii in ticks are wrong, because molecular biology helped us recently to discover that there are some Coxiella-like endosymbionts, which are Coxiella close to Coxiella burnetii, but not pathogens. And there are some bacterial agents living together for 1,000 years with ticks, with the co-evolutions, but maybe they were confounding this Coxiella-like endosymbiont and Coxiella burnetii. So probably that many papers from on Coxiella burnetii in ticks are wrong, and there are some other Coxiella in ticks without infections, but it is not the point. And after the wildlife cycle, the bacterium goes to the domestic animals, and especially livestock, and small ruminants are the most implicated animals in the human epidemiology. there is a consequence on the animal health with consequence of the productions of the babies in the livestock. Humans is contaminating through the dust and through the excretion of the bacterium in the environment through genital matrix of females from goats and sheep. So there are some specific population at risk, such as farmers, veterinarians, and slaughterhouse workers. In French Guyana, sorry for this ChatGPT production, we contributed to the understanding of the transmission, because Q fever is supposed not to have any human-to-human transmission, but we had a case of human-to-human transmission from a French Guyana case a few years ago, with a couple going to New York, US, in the winter, and meeting there the brother of the woman. He was coming from Canada in the middle of the winter, so there is no transmission of Coxiella burnetii in Canada during the winter. And the man from French Guyana got sick at the arriving in New York, with cough, very important, during 1 week in the hotel room with the other guy, and the other guy developed a Q fever at the return in Canada. So this is a possible, not proof, but a possible human-to-human transmission of Q fever in between two humans. And this one is just a joke, but I supported a patient a few years ago, and the story was very funny because he met a three-toothed sloth, which is very common in French Guyana, on the road. So he helped the animal to get away from the road, and the sloth scratched him. So ChatGPT didn't want to show what I wanted him to show, but the guy who was a little bit special introduced his finger in the ass of the sloth because he had scratched him, and a few days after, he developed an infection of the finger, and a few days after, he developed a Q fever pneumonia. So there is no proof, but we've got someone with a contact with the sloth, who introduced his finger in the bad hole and developed a Q fever a few days after that. It's just a chronological link, but well, the contribution of French Guyana in the knowledge of the transmission of Q fever. Let's get more serious. Q fever, infection by the inhalation of the infected dust, 60% of asymptomatic infection. And in general, 10 days to 20 days after that incubation period, people develop high fever, headaches, and in some cases, they develop liver inflammation, hepatitis, and sometimes, and especially in French Guyana, pneumonia. And in general, it's a self-limited diseases, 2 to 3 weeks of fever, and then even without any antibiotics, the people get a good evolution. Some people, especially people with heart valves problems and immunosuppressed patient, may develop chronic Q fever, also called persistent chronic Q fever, and the main clinical picture is an infective endocarditis and vascular infection. Which is the main burden of the infection. The diagnostic relies on the serology, but only possible 7 to 10 days after the onset of the symptoms, before the antibodies will get negative. And we are showing actually that PCR in the blood could be a very interesting tool at the acute phase of the acute Q fever. The diagnosis of the chronic Q fever, persistent focalized Q fever, is made on the IgG, phase 1 IgG serology, and also the PCR on blood and tissue. I won't talk about the treatment today, that is not the point, but it relies on the doxycycline and antibiotics in the chronic form with on doxycycline and hydroxychloroquine for 1 to 2 years. This is quite a rare disease, which high variable prevalence from one country to another. For example, the history of the Netherlands was not really marked by Q fever until 2007, when they had the uh, highest cluster, the highest epidemic of Q fever, highest outbreak of Q fever reported ever in the world. And they had something like more than 4,000 cases within 3 to 4 years, and linked to goats. But in general, you've got some small focus all around Mediterranean Sea, and also in Canada, the US, etc. And probably, and I will talk about French Guiana, but probably there are some places in the world where it is very underestimated, and I'll show you a slide on Africa. So it is supposed in every paper it is written that there is a complete worldwide distribution except New Zealand. In all the papers it's written that, but when you I will show you another. But when you looked synthetically to the medical literature, for example, for Africa, this is something I presented in Guinea a few years ago. There are some countries that never described Coxiella burnetii in humans or animals, but it's supposed to be everywhere. In Latin America, we published a paper in PLOS, Neglected Tropical Diseases, 10 years ago, and we showed that at least 5 countries never described Q fever in humans or animals in the medical literature: Belize, Chile, Guyana, Honduras, and Suriname, and some of them for more than 30 years without any case. Let's talk about the very specific features of French Guiana epidemiology. As many infectious agents, it was described for the first time in the 50s by Hervé Floch, which was the director of the Pasteur Institute of French Guiana, and he found Q fever in humans and cattle for the first time in the 60s. But between the 60s and the 90s, nothing special happened for Q fever. In 1996, this is a publication in Emerging Infectious Diseases, 3 admissions were made in the intensive care unit for acute respiratory distress syndrome with 1 death. So they started to be interested in this special disease, which has really not been described before. So they've made a first study on all the sera sent to the Institut Pasteur for microbiological diagnosis, especially for dengue fever. And so they performed the serodiagnosis. And the first interesting thing showed that between 1992 and 1996, the positivity rate to Coxiella burnetii in the sera grew very quickly from 2% in 1992 to 24% in 1996. So another retrospective study was performed after that. They investigated all the cases of Q fever between 1950 and 2006 in French Guiana, investigating all the lab results in French Guiana. Between 1950 and 1999, only 10 probable case, but 1 single live, the IPG for Institut Pasteur de Guyane. So nothing very relevant. Between 1990 and 2006, almost 1,000 cases of Q fever were described, as you can see on this incidence curve, with something starting in the middle of the 90s and with a very high incidence increase in 2005, and I will show you. For you to understand the real burden of this, the importance of this incidence rate, let me show you. So 19 incidence rate, almost 2 cases per 100,000 inhabitants per year. And until 2005, 120 cases per 100,000 cases inhabitants in 2005. At the same period in France, in the National Reference Center, France, which is supposed to be a country where the disease is sometimes described, the incidence was 0.32 cases per 100,000 inhabitants. So to 120 to 0.3, there is a factor of 500 more frequent cases of Q fever in French Guiana at the same period. Even comparing to other countries such as Australia, Taiwan, etc., where Q fever is often described, there was no comparison with this very, very, very high incidence. A few days ago, I put some photos of people I worked with during all these years. And this is Pauline Thiel from Lille, and with her, where she was resident in our department, we performed an incidence study of Q fever between 2007 and 2017, and we showed that between 2010 and 2017, the incidence was still very high, with 40 cases, 27 to 40 cases per 100,000 inhabitants per year. So one of the highest incidents in the world. That's why we call this paper the hyper endemicity of Q fever in French Guiana. When you show the incident between 19 and 2017, that gives this evolution curve. So another very strange specificity of Q fever epidemiology, human Q fever epidemiology in French Guiana, is the location of the cases, because most of the cases are located around Cayenne, which is the main city of French Guiana. The first study in 1996 showed that most of the positive sera came from Cayenne versus the rest of French Guiana. Another study performed by Carole Eldin a few years ago showed that between 2008 and 2012, that was the same phenomenon. What we call the Cayenne Island, with the other municipalities of Remire-Montjoly and Matoury and Cayenne, there is a significant difference of the incidence between Cayenne and surroundings and the rest of French Guiana. Sorry, I think that one slide disappeared. Oh, okay, sorry. One of my slides disappeared, but in the study performed with Pauline Thiel, published in PLOS Neglected Tropical Diseases, we showed that between 2007 and 2017, 85% of the cases occurred in Cayenne and surrounding. So a phenomenon still going on in the recent years. This is another study performed by Sarah Bailly here during her thesis, and with her thesis director, Claude Flamand. And they showed this is a seroprevalence study. Thanks to a seroprevalence study on arboviruses in French Guiana, more than 3,000 samples of blood in all the municipalities of French Guiana. And then they use serotech to study Q fever seroprevalence. And they showed that always Cayenne and surroundings were one of the hotspots of Q fever. As you can see here, 17% of the population of Matoury, 9% of Cayenne population, 17% of Remire-Montjoly population. So here are positive to French Guiana. And also some towns where there are very few cases of Q fever described, but with where the seroprevalence is very elevated, such as Cacao, close to the shore. let's talk about clinical aspect of the Q fever. What we can show about the clinical aspect of Q fever in French Guiana is that most is the high pulmonary tropism of Q fever in French Guiana. I already said, but most of the cases described anywhere in the world, in general, it's isolated fever, hepatitis, and depending on the study, 5 to 40% with pulmonary involvement. In French Guiana, there is something very special. This is a study not published, but presented in our infectious disease congress in 2007 by Tanya Vaz, which is here, and that shows that 90% of the acute Q fever cases were pneumonia. This publication by Sophie Edwards from the Marseille team and Abba Mahamat, which was working on Q fever in our department before going to course, showed that on the 2008 to 2011 period, 8% of the cases of Marseille area were acute pneumonia versus 83% of the cases in French Guiana. So a real difference. There are some bias in this study, but a real difference in the clinical presentation with a pulmonary tropism very important. This is when I got into the Q fever story. I was resident in the infectious disease department of Professor Félix Jossou in 2007 with my colleague Cédric Chenet, and we performed a study on a 4-year study on acute pneumonia admitted in the department in the Cayenne Hospital. So we showed that among 131 acute pneumonia hospitalized and admitted in the department, 24% were due to Q fever, something never showed elsewhere. Carole Eldin, which is an infectiologist in Marseille, which has resident after me in the department, performed the same study between 2008 and 2012, and she showed that almost 40% of the admitted community-acquired pneumonias were linked to Q fever. Just for you to understand, this is the comparison we made in our publication in 2022, and we showed not all of them, but many series of the on community-acquired pneumonias all over the world, in the United Kingdom, Western Europe, Netherlands, Spain, Israel, etc. And in general, Q fever represents 0 to 1 or 2% of the big series, compared to 38% of our series in French Guiana. So there is a real issue on Coxiella burnetii community-acquired pneumonia in French Guiana. And what's the point? Until 2014, so almost 20 years after the discovery of the importance of Q fever in the human medicine, there were still no idea about the specific strain of French Guiana. And fortunately, thanks to Abba Mahamat and the Marseille team, 5 strains of Coxiella burnetii from Cayenne patients were analyzed in the National Reference Center, and they showed that they were all due to the same strain, which was called MST17 for multispacer sequence typing. And the very interesting thing is that all the strains identified in French Guiana after that in the further years were all due to this unique clone of Coxiella burnetii, MST17. On the contrary, when they studied all the bank of the National Reference Center of France in Marseille, 100% of the MST17 cases were found in people who were living in French Guiana or who had traveled to French Guiana. So there is a real unique genotype linked to French Guiana Q fever. Cléa Mélénotte, which is a French researcher. She was also resident in the department 10 years ago, and she worked for a few years with Professor Raoult and Marseille team. published this very interesting paper where she studied our French Guiana strains compared to the 9-mile and German strains, which are the reference in microbiology of the Q fever. She showed that our strains is hypervirulent, including in silico, in vitro, and in vivo, compared to the reference strains. Another very specific aspect is the ethnical distribution. This was not published because you know that it's quite difficult to publish ethnical statistic. But in the French, in the first studies we performed with Cédric Chenet, published in Clinical Infectious Disease in 2012, we showed that 60% of the people with Q fever, with pneumonia linked to Q fever in the department, were from mainland France. Just to remind, that was the Q fever group, that was the community acquired pneumonia not linked to Q fever. So the control group, where you can see the multi-ethnicity of French Guiana for pneumonias, mainland French, Creoles, Brazilians, Haitians, Maroons, Amerindians, Suriname, Guyana, etc. So there was an over-representation of people from mainland France, metropolitans, as we call them here, in this first study. In the second study we performed with Pauline Thiel, you can see that among 700 cases of Q fever in French Guiana between 2007 and 2017, most of them were from French Guiana, but the second ethnic group were mainland French. If you compare in this table to the general population, you can see that 30% of the cases linked to Creole people born in French Guiana, it's the same than in the population, but there was a real strong over-representation of people from mainland France in the cases described on this study period, and an under-representation of people from Haiti. And when we compared the 700 cases to the general population, we showed that the risk factor were an age between 30 and 60 years old, male gender, place of birth, mainland France, and place of residence, Cayenne. Another study was very interesting, the same one of seroprevalence on the collections of blood in 2017 from Claude Flamand and Sarah Bailly, and showed that the most prevalent ethnic in French Guiana were country born in French Guiana were people from Haiti, which is really different from the acute cases, which were more from people from Europe. So there is another query things about human Q fever in French Guiana. And the last part of my talk, I'm supposed to have, yes, it's 9 and 5, so I've got time. So one of the very mysterious query things of Q fever in French Guiana is, what is the animal reservoir? As I told you, the group at risk in the rest of the world are veterinarians, are farmers, are people working in slaughterhouse. Nothing to see in French Guiana. A really interesting study published in 2001 where they compared people with fever with positive sera to Coxiella burnetii versus negative sera to Coxiella burnetii. This is the result of the multivariate analysis, and it shows that people working in building trade or public works were one of the most important at-risk group. People seeing bats in the house, seeing wild mammals, proximity to forest. People who know French Guiana know that it's not so strange to see bats or wild mammals near house. But you can show that there is no link to farmers, to veterinarians, and or a rural point in this group at-risk groups. And in this study, they captured some animals, and there was no PCR at the time, but they found some antibodies in some marsupials, quite common in French Guiana, and one wild rodent, without knowing if there was a link or not. Vincent Pommier de Sainty, which is a military physician and epidemiologist who worked for years on French Guiana, on Q fever in French Guiana, while he was working here, made this review of the literature of the knowledge on Q fever in French Guiana in animals. And as you can see here, nothing found in the livestock in French Guiana, even in cattle, sheep, goats, porks, nothing. Some very, very low positive sample in dogs and something in wild fauna, but nothing very clear. And also in French Guiana, there is a myth and people thinking that Q fever is linked and is due to bats. And I don't really know where this myth come from. But as you can see here in a local paper, sanitary problems caused by Q fever. We don't really know the origin, the root of this very fake news. One of the turning points in the knowledge of the animal reservoir or Q fever in French Guiana is this cluster in a place called Camp du Tigre Tiger Camp, which is a small hill close to the French, to the hospital of Cayenne, and which was residence for militaries, soldiers, and their family in the middle of the forest on a small hill. And between December 2012 and June 2013, they had 11 cases of Q fever among 54 people living in this place. Half of the households were affected. And among them, three people had positive PCR and always MST17 identification. So one of the consequences is that all this area was closed to the public. And so today it looks like Cambodia, Angkor Wat, with buildings invaded by and surrounded by the forest. And so Vincent Pommier de Sainty made the investigation, and he showed that the activities linked to the sick people with Q fever were the people who had cleaned the house, so maybe with the dust in the environment, and to hold three-tooth sloth in arms. That's something very interesting because all the Marseille team came to investigate this cluster. They captured hundreds and hundreds animals on the Tiger Hill, and they found a dead sloth on the road to the building, and they analyzed it, and they found for the first time a positive animal to Coxiella burnetii with the same strain MST17. That was the first time that an animal was positive for Coxiella burnetii in French Guiana after 25 years, almost 30 years of cases in humans. Well, after that, the team wrote some not completely true things in various papers, saying that the epidemics of Q fever in Cayenne and French Guiana was related to the opening of a shelter for three-toothed sloth in the city center, highly visited due to the interest of the population. People from French Guiana don't really care about sloth, so I don't really understand that. So it seems that the epidemic of Q fever of Cayenne is linked to a wild animal reservoir living in very close proximity with the inhabitants of the city of Cayenne. All the people in French Guiana reading that say that's a very strange hypothesis.
Loïc, you have 5 minutes left. 5 minutes.
Okay. So just to tell you, the shelter is here, Shuai, so 8 kilometers from the center of Cayenne. So it's not in the center of Cayenne. All the sloth sampled in Shuai were all negative. for Coxiella burnetii. So when I read that, I wanted to go a little further to understand the animal reservoir in French Guiana. So that was there was another cluster in militaries, and we showed that capybara, which is the biggest rodent in the world, was this cluster was linked to these specific animals. They had used a brush cutter, and there was some tools of the animals close to the house they were living, so that could explain this small cluster of 5 militaries. So we wanted to know a little bit more. Just for you to know that there are some more recent studies that do incriminate again livestock in the epidemiology of Q fever in French Guiana, because they found a link between high seroprevalence and the distance to farms, but that is a statistic link that we can't completely explain. Another study from Mona South that showed that maybe the seroprevalence in livestock in French Guiana is higher than in the rest of France. Also, a study we performed with Anissa Demoulin, which will be published this week normally, showed that there was a link between the slaughterhouse of French Guiana and the cluster of the cases in French Guiana. And wildlife. This is something, a study we performed recently in French Guiana to try to understand which species are incriminated in the epidemiology of Q fever, human Q fever in French Guiana. So we wanted to identify mammals incriminated in the epidemiology without killing any animal, because most of the studies on Coxiella burnetii in wildlife in the rest of the worldwide literature, they kill bats, they kill rodents, they kill animals to study Q fever, and we didn't want to do that. So we had to think about where to find some samples without killing any animals. So first, we made some bat captures with what we call Japanese net. The bat go into that, you take it out from the net, and then you use the tools in the bags where it is having rest, and then you make some samples, especially samples of rectal and vaginal samples in these animals, and then you release them. We went to the zoo, which are in the middle of the forest, and the rescue centers to make some samples in various animals, monkeys, wild boars, tapirs, etc. And then we used the French Biodiversity Office collections, because they have a high collection, a huge collection of tools from jaguars and various herbivores. One sample of sloth on the same place that Marseille team found on the hill, Tiger Hill. And we also used a collaborative study to the population, saying if you find a dead wild mammal on the road, call us. So we had some wild killed animals such as tattoos, rodents, one jaguar, etc. And pre-existing collections from the Pasteur Institute in French Guiana. So we made the extraction, specific PCR for Coxiella burnetii and for the French Guiana strain. So what we found among 2,000 samples from more than 10 orders of mammals, Artiodactyla, Carnivora, Chiroptera, Marsupiala, etc. So 200 swabs, 500 stools, urines, tissues from dead animals. And we found that was very interesting for us. These results are very extraordinary. 34 positive samples from their various samples, stool, anal swabs, kidney, liver, lung, etc., form 16 different species of wild mammals in French Guyana. 3 of them could be analyzed, and that was the same strain MST17. So we had wild boars, kinkajou.
Loïc?
Ouais, c'est mon avant-dernière diapo.
Yes, perfect.
Jaguars, 2 species of bats, 2 species of marsupials, including this one, which was very prevalent in the human habitations everywhere in the cities, but also 3 species of monkeys positive, and at last, positive samples for Rattus rattus and Rattus norvegicus, so black rats and brown rats, which are known for 3,000 years to be the vectors of various diseases such as Yersinia pestis, leptospirosis, etc. All right, then, I'm going to stop here. There are so many other projects, studies on birds, on rodents, also studies on human Q fever to better understand the epidemiology, the location, to know how is the incidence, because there is, as it's not a mandatory disease, we don't have any idea of the incidence between 2018 and 2025. And all the people who helped me to perform the Fornacock study. And sorry, I'm not so late. 9:17, it's perfect. So thank you for your attention. And if you have any question, I see that in the chat there are many questions. So let's see how we can manage that. Thank you so much.
Thank you, Loïc. Thank you for this, for sharing these results, not of 20, but 30 years of research of Coxiella burnetii in French Amazonia. It is really impressive to see how you and your team, how far you and your team have come with this research and the results. As you mentioned before, we have several questions in the chat, so I won't take time because I want the audience to ask the questions directly. So we have 4 questions pending in the chat, and I see that someone is also raising the hand, so this is really an exciting moment for you to share with your audience. So we have the questions asked in French. We have the first one was bonjour, parmi les symptômes de la fièvre Q, les humains, parmi les symptômes chez les humains, vous n'avez pas évoqué la tente hépatique. Or, nous l'avons observé en région Centre-Val-de-Loire suite à un contact avec un chevreuil naissant. Okay, ça, c'est la question de monsieur Henri Taboul.
Thank you so much for the question. So in humans, in the European form of Q fever, this is the main involvement in humans. It's a fever with hepatic liver involvement. But this is something we don't really have in human cases in French Guyana, but in the rest of the world, of course, the liver involvement is important.
We have another one. How long can a person or an animal who has been in contact with a bacterium continue to have detectable antibodies? In other words, after how long is it no longer possible to determine whether a person or an animal has previously been exposed to the bacterium?
Yeah. Normally, the antibodies are present for the entire life. That's why we can make all these seroprevalence studies, is that years and years after that, we can find some antibodies when we make seroprevalence studies. And also, Q fever is supposed to be an immunizing disease. So if you got Q fever once, you're not supposed to make the infection again.
We have another question. Have there also been similar studies conducted in other countries retrospectively in order to truly compare the incidence rates?
Yes, of course. Not really in the South countries. the research on Q fever in the rest of South America is just starting, except in Brazil, where then they have some publications, but not so much, and there are no incidence rates. It's in no countries. This is a mandatory disease, so we don't have really incidence rate. And otherwise, we've got some. We have some results from the National Reference Center in Australia, in France, in Israel, in Switzerland, in the Netherlands, but the rates are not so high than in French Guiana.
I have another question then. Could the higher incidence rate in large urban centers be linked to a higher rate of diagnosis? Are there few consultations for fever in Ireland rural communities?
Maybe there's some bias, but no, we studied that point, and the rate is higher in the. We don't have big urban centers in French Guiana, but we've got some cities, and no, the rate is higher in the urban parts than in the rural parts. We studied that, but you are right on the point. Is that there is no investigation. There is not so much investigation on Q fever in people with fever in the remote towns of French Guiana because of problems to bring the blood samples from the remote centers to the lab in Cayenne. I think that Solène have got to ask question.
Yes. Would you like to turn on the camera and to ask the question directly?
Hello, but no, there are other questions in the chat, so I didn't want to.
I think that there are some other questions.
Yes,
otherwise, I have a quick one. I'm Solène Kernis. I'm the coordinator of the graduate school. And so thank you so much, Loïc and Paola, and Loïc for this fantastic talk. I'm very happy to finally have had the opportunity to listen to it. So thank you so much. I was very interested on the route of transmission because the fact that you mainly have pneumonia, which is quite different from what we usually observe in other places. And also the fact that you have found that being exposed to dust is actually a risk factor to be infected. And when you perform samples, have you ever tried to find Coxiella in the environment? I mean, on surfaces, in the dust, aerosol, because sometimes you can capture microorganisms in environmental samples.
Honestly, this is one of my projects, but I've got so many. So this one is one of them with the National Reference Lab in Sophia Antipolis. And they already performed that kind of study when they've got some clusters in farms in the south of France with samples in the environment of the farms. I did that once during the COVID period because we had a couple, one man and woman. They didn't get out from their house for 3 weeks linked to the COVID, and they both developed Q fever. So I went there with my team, and we made many samples in the environment, but everything was negative. So in French Guiana, that was made only once and nothing. And there is another study, another team which wants to make some investigation around the farms, the cattle farms, but I don't think it's already done.
Thank you.
But for example, I didn't show this publication with Timothée Bonifay, was one of my colleagues. We made a publication showing that many people from the jail, from the prison of Rémière-Montjoly in French Guiana, had a cluster in 2017, 2018, and they were in jail, so they didn't get any contact with the fauna or with the nature. So probably that was linked to the very resistant bacterium in the dust and so they inhalated infected dust and got sick in the prison.
Yes, it really plays for aerosol contamination and also the clinical form.
And for the pulmonary tropism, that's not the point today, but we've got many infectious agents in the Amazonian area, which are not specifically pulmonary all over the world. And here, they are pulmonary. For example, toxoplasmosis. Acute toxoplasmosis here gives interstitial pneumonia. Hantaviruses, which are in general with liver and kidney involvement in the old world. In the new world, it's especially pulmonary involvement. Leptospirosis with very frequent pulmonary involvement, Q fever, etc. So there is something special maybe with the humidity of the environment, which gives a special tropism to the lungs for our infectious agents.
Well, we have some other questions in the chat. So do not hesitate to turn on the camera and to ask the questions directly. So it is very interesting that all the Coxiella burnetii from French Guiana belong to MST17. Is there any particular features of the MST at the genomic level?
That's not so my specialty, so I couldn't really tell you. I can just tell you that it's been studied by the Marseille team, and there are some probably specific virulence gene, but I can't really give you a satisfying response.
Well, next one. So, bonjour Loïc. This is LO. So, LO, sorry, I don't have your full name. Bonjour Loïc. Qu'en est-il de la fièvre Q chez les enfants?
That is what we call a trap, because
Yes,
LO is a pediatric infectiologist who was in French Guiana for 10 years, so he knows the response. Q fever is supposed to be almost absent in children. There are some small studies from Israel with chronic forms, but very few things. So Lindsay knows that we are starting a study in the whole France to collect all the human cases in children. In French Guiana, we found in the last 10 years something like 30 cases, but we could make a description of 17 cases, and this is like just fever, sometimes cough. And there is also a study from Kenya, I think, they show that children with Coxiella burnetii antibodies have more fever and more cough than the other children with fever without any Coxiella burnetii antibodies.
Thank you. So
Thank you.
We have another question. Comment distinguer les symptômes souvent non spécifiques avec les symptômes de la dengue, la grippe ou la COVID-19?
Thank you, Zaina, for the question. It's more difficult than that because you're talking about infectious agents, but we have other here. Very difficult to make a distinction from dengue, influenza, SARS-CoV-2, and also malaria, which is endemic here, and also leptospirosis. So it's very difficult. The first publication I made a few years ago, I showed that there are some small specificities of Q fever. For example, to distinguish from leptospirosis, you've got very high C-reactive protein with normal white blood cell counts. What else? To make the difference from dengue, you have also the C-reactive protein, which is not so high in dengue. From COVID-19 and influenza, the pulmonary involvement in more than opacities such as Streptococcus pneumoniae than interstitial pneumonia, as you can find in influenza or SARS-CoV-2. But of course, it's quite difficult to make the distinction, and we need our lab and our biologists.
Et enfin, nous avons le temps pour une dernière question. So we have time for the last question, I think. So it is related to the enfin, quelles sont les caractéristiques du clone amazonien MST17, MST117 par rapport aux souches européennes et africaines?
So we can't talk today about the Amazonian clone, the Amazonian strain, because it has only been found in French Guyana. In the other countries, it has never been found, except maybe, I think that it was found once in a Brazilian study. But the characteristics are that, as described by Clea Melinot, it is more virulent, in silico, in vitro, in vivo, in the rats. So compared to the known strains, so 9 Mile and German strain. But there is no possibility today to compare to the other American or African strains, or it's not been done yet.
Well, I think it's time for us to close this incredible session. Just before leaving, I want to tell you that Professor Kenneth is sharing with us in the chat the website of the graduate school, and I'm also sharing with you the link to the videos that are already online. So please go to check our website, the graduate school website, and also the media section to check on the previous videos who are already posted, the videos of the previous sessions. Well, Loïc, yes, go ahead.
I'm sorry. Thank you so much, Paola. Maybe you can say one word about the social sciences seminar webinars which are coming.
Yes, of course. So we are also having the other series, which are the social and human sciences series of the graduate school, and that will start on March the 25th. So they already started. The sessions already started last November. We already had Freya Jepkot's presentation on January, and we will have next presentation on March the 25th. So I will share with you also all the information on the program soon. This series is being organized by the postdoctoral researcher Jules Villa from Pasteur Institute.
And so thank you very much again. And next talk, who is it? Loïc and Paola? What have you prepared for the next session?
Jean-Bernard
Duchemin.
Okay, and about...
You will talk about mosquitoes and the research of mosquitoes and arboviruses in the French Amazonia.
Fantastic.
Well, thank you all for joining us, and we'll see you next month on March 18 for French One Health Research in French Amazonia, and on March the 25th for Social Sciences webinar series. Bye.
Bye. Thank you so much.